So something that you all should know before you jump into picking your topic is what genetic transformation is, or genetic engineering. So how we kind of do this is, the first thing we need to do is take a plasma, which is the DNA, a circular DNA, from a bacteria cell and remove it. Then we're going to cut it with restriction enzymes. You can think of these enzymes as scissors, and they're going to look for this palindrome that are a sequence of DNA that they are going to cut it with. So the first thing is to do that. The second thing is, then say we want to take a gene from a jellyfish, and we want to insert it into something else. So this is this PGLO gene, and it's antibacteria resistant. So that's why we want to put it into, or make more of it, or put it into another organism. So we're going to take the gene of the specific place in the jellyfish that we want. We're going to cut that with the same restriction enzyme, and we're going to pull it out. Now we're going to combine the plasmid DNA of the bacteria cell and the jellyfish DNA. When we put that together, it's called recombinant DNA, or rDNA, which means that you have now recombinant, you have two different types of DNA. Well, they're all DNA, ATCGs, but you have two different organisms of DNA that are combined together. We're going to take that recombinant DNA and put it back into step 4, put it back into the bacteria cell. Once it has it back in the bacteria cell, again, the bacteria cell doesn't know that it is not its own DNA because it's all made up of ATCGs. So it's going to go about its normal business, doing its thing, replicate, making that protein. So the glowing gene that is spliced in there is actually going to be made by, that protein is going to be made by the bacteria. So it's going to continue to divide and grow, and it's going to have this gene in it. And when those genes get turned on, the bacteria cell will also glow. So question, why might it be important for the same enzyme in step 1 and step 2? So remember that we had in step 1 the plasmid that was cut by an enzyme that was looking for a specific DNA sequence, and we had the same one that was cut over here with the DNA from the jellyfish. Well, it's really important so that the plasmid and the glow gene have the same sticky ends that match up. So because they're being cut by the same DNA, or the same enzyme, they're being cut at the same sequence of bases. So for example, if it was cut here at AATT, right there, that sticky end would also pair up with a sticky end that has the matching base pairs of the plasmid. So if different enzymes would use, then they wouldn't stick together. There is more than one restriction enzyme, and we use them for several different things. This process worked on a bacteria cell, but would it work on larger organisms like humans? Why do you think? Why or why not? Remember that a bacteria cell is really small. It's not going to work really easily because we have trillions and trillions of cells that would have to be changed, but because bacteria cells just have one, then it's real easy to do that and make lots of that protein. Then we can extract that protein to use, such as, say, insulin or human growth hormone, something like that. If we're going to be using this for something for gene therapy, we would actually use something like a virus, because it would be easily accessible to infiltrate our own cells at a much faster rate and change over just the cells that we need. All right, tell me what it is that you're interested in for yours. If you're interested, or if you need to know more about some of these vocabulary words, here is what each of them are that we just went over, and I can't wait to see what you're interested in to do for the next couple of weeks.